All right, so it's my pleasure to welcome you to today's seminar that's being given by Professor Margie Mulholland from the Ocean and Earth Sciences Department at Old Dominion University. Dr. Mulholland has a diverse background in biology, public policy, a whole number of different things, and starting out with a bachelor's degree in geology and biology. She has been working for a number
looking
of
at the
years
biology and
in
ecology of
harmful
harmful
algal species. She is currently the lead PI on a NOAA-funded monitoring and event response for harmful algal blooms project. Her seminar today is going to be related to that, but setting up, we'll learn here about how you monitor for that and then how you would use that information to input into potentially forecast models. So the title of her seminar today is Enhanced Surveillance to Improve HAB Monitoring and Detection Toward an Early Warning System for HABs in the Lower Chesapeake Bay. So Margie, please go ahead.
All right. Well, thanks everybody for coming on this miserable day to go outside. We were actually out on the water this morning before it got nasty out. So, but thanks everybody. I have listed here a group of the co-PIs on this particular project, and these are just the funded ones. There are plenty of other partners that I'll show you at the end that are not receiving funding from this. And we're very actively engaged with NOAA and the Mid-Atlantic Observing System, the Maricus, on this project as well. And so, oops, it's not going down. Let's see. There. Okay. So first, to provide some context for you all and get you to where we're at now, because this has been really, since I've been here, I've been working to try to get this or something like this going. And over the last 50 years, bloom-forming dinoflagellates have become increasingly abundant in the lower Chesapeake Bay, and they bloom. They've been linked with nutrient over-enrichment, and they can form very, very dense blooms. Some of them are directly toxic, or they can also be harmful by exerting negative ecosystem impacts. There's specific species, that's hard to say those two words in a row, occur with regular seasonality, and we're trying to understand why that is. Are they filling some realized niche, or are they in their optimal niche? We think probably the former, actually, for many of them, but they vary from year to year. And there are monitoring programs aimed at assessing water quality and impairments in the Chesapeake Bay. You probably all heard of the Chesapeake Bay Monitoring Program that does monthly monitoring. They were supposed to be out today, in fact, and they collect samples at 14 stations for phytoplankton analysis from the main stem. There's 7 stations in the main stem in Virginia and 7 in the tidal tributaries. So there's many, many more water quality stations, but the ones at which phytoplankton are collected are limited to 14 because it's really quite a laborious analysis to analyze these phytoplankton samples. There's also in yellow, the yellow dots are shellfish sampling stations that the Department of Health collects monthly from 69 stations. And in those samples, we just count HAB species. We don't do the full population assessment like we do for the Bay program. Then there are also, VDH also has toxic stations where they look for toxic algae and they measure toxins, and these are monthly as well. And then there's ad hoc bloom reports that we'll get samples in. If someone sees discolored water, we always encourage them, grab a sample, bring it to us, and we'll analyze it for you. And the Department of Health has a hotline where you can report discolored water, and then they try to send someone out and get a sample, but can't always happen. But as I indicated, different bloom species, they vary seasonally. So when you get a bloom in the winter, it's usually Heterocapsa. When you get a bloom in the summer, it's usually Margolefidinium. So they're just different taxa that have different optimal temperature niches. And so this is data collected between 2007 to 2020 that we used to build this figure that we published in a 2018 paper here to look at the thermal window in which these different potentially harmful species occur. And so but we have many different HAB species in the bay, and so it's not something that we can distinguish between the species optically or any other way, because we just have to look at them under the microscope, which again, like I said, is very laborious. The blooms that we're focusing on for our MARHAB project, of which Eileen's a part, is we've been focusing on Margolethidinium polycricoides, or Marge, and then Alexandrium monolatum, and we call them Marge and Alex for short, because the full species name is a mouthful. But these blooms occur in the late summer when the water temperatures are warm. The blooms of Marge were first observed in 1994. We think they entered the Chesapeake Bay from some other waterway. We don't know where, but they were first observed in 1994 by Harry Marshall. And then blooms of Alexandrium didn't emerge until 2015 in the bay, in the lower bay. Or I'm sorry, maybe, 2015. But here you can see the red dots over here. This is the observations of blooms of Marge between 1994 and 2003. And this is blooms of Marge and Alex now from 2004 to 2013. So I guess they were here in 2013. And then you can see that they've expanded substantially in the region. So, and this is between 2014 and 2023. And yeah, and in recent years, I'll just show you a couple of slides. This talk is really an amalgam of several presentations that I've given over the years. But the Alex blooms, we think they started emerging after Marge, perhaps due to some shift in climate or some expansion of the thermal window during which their blooms occur in the lower bay. In Virginia, Margolethidinium likes it hot, so they bloom when temperatures are hot. However, this is not their realized niche everywhere in the world or even in the country, because up in Long Island, they occur where salinities are much higher and temperatures are lower. But yet they're recognized as being genetically identical. But blooms have been emerging worldwide. It may be in response to expanded monitoring, but it's more likely a response to that they're increasing around the world. But these blooms can be spectacular. If you've ever crossed any of the bridges, this is just Hampton Boulevard here, and we have a time series station here at the Yacht Club. And so you can see that these blooms are quite vast and they're very patchy. So sometimes monitoring is insufficient to sample them just because you're sampling at a fixed station and you're sampling in a light colored area versus this dark colored area. So it really sometimes matters even what side of the boat you sample out of, whether you are able to get them in your samples. But the cell densities can reach tens of thousands of cells per milliliter. And Alexandrium is always, people take notice of it because it glows in the dark. It's bioluminescent. And so it made the front page of the newspaper one year when it was blooming. And actually, I have a funny story. The NPR wanted to interview me during a bloom of this, and they wanted me to come to the site. And it's like, you're on the radio. Why am I going to the site for an interview? And he's like, I want to set the mood. So anyway, I thought that was pretty funny. The other thing about dinoflagellates blooms is you saw that they were patchy, but they also vertically migrate. At the Yacht Club, at our time series site, we actually keep a mooring at the top and the bottom, so we can see that this migration over the course of the day. But these three pictures are just pictures of the water taken at 3 different times of day. And so in the morning, it was only 21 micrograms per liter of chlorophyll, but by midday, it was up to 530 micrograms per liter. And cells don't grow that fast, but they had just migrated to the surface from the morning to mid-afternoon. And so these, you can sometimes smell the algae too when they get this thick. But that's just a picture taken at the Yacht Club. So we essentially built this conceptual model of a bloom, which is essentially a growth curve. You have bloom initiation when cells are just beginning to double and increase, and then you have an exponential growth phase where the population is growing substantially. Then there's transport and maintenance of the bloom, so it just sort of keeps going, and these blooms can last for months. And then there's a point in time when they decline. And this all just reflects a balance between population growth and loss. And that is dependent on things like nutrient availability, light availability, mixing, grazing, microbial interactions, viruses, and different things. And then the population transport, and I'll show you this later, but we think that blooms initiate in specific areas and then are transported around the bay. And they are transported by tidal advection, the estuarine circulation, wind-driven flows, and river flow. And I'll show you results from a model about that a little bit later. Most studies are done once a bloom is visible because people see discolored water, so you go out and get a sample. But the million-dollar question is actually what causes these blooms to initiate? Because we know for Marge, once they initiate, they're scrappy little cells. And so they can exist they get transported around and then they can thrive for long periods of time. So this was the million-dollar question for the grant we had that preceded us getting the MARHAB grant. It was an EcoHab grant when we're just learning more about the species. So first problem we had to solve was to find a site of initiation so we could sample that, so we could learn more about why blooms initiate, what conditions get the population going. The Bay program sampling is monthly, so that's not going to be frequent enough, right? And the conditions during a well-developed bloom or at that maintenance stage of a bloom are very different than those during bloom initiation. And as I had said, blooms are patchy and move around, so we need to find those sites of initiation so we can stop the blooms from happening. So we tapped into an HRSD. They do water quality monitoring as part of their permit. They do something called data flow cruises, and they do it in these two segments of the James River down here in the polyhaline and the meso and oligohaline areas of the James River. I'm going to show you some plots, and so these are going to get stitched together in a weird way in some of the plots I have. But they do these cruises biweekly between April and September, and they essentially put a YSI data sonde on a boat and flow water through it. And these data sondes measure chlorophyll A, salinity, temperature, dissolved oxygen, pH, and they've been doing this since 2005. And we convinced them to go into the Elizabeth and Lafayette rivers between 2008 and 2015. And this is just what that looks like off the back of their boat. Okay, and I said these are going to be stitched together in weird ways, and that's because these maps are actually connected right here with the upper part of the James River in here. And they do it this way because they sample this segment one day and this segment the next day. And so what we did is we looked at the data flow data that HRSD had, and this is from 2007. These were old data that we looked at. And what we found was that these are chlorophyll fluorescence. And here you can see that this area in the Elizabeth River sort of lights up first. And up here, there's a couple areas that are lit up, but they're not massive blooms yet. And so this was on August 7th in 2007. And then the next week when they went out, there was much more chlorophyll in the Elizabeth River and then across the river. And again, not too much in the oligohaline and mesohaline, but by the next week, it was spread throughout the lower James River and above. And so we speculated that the Elizabeth River was the source. And so we asked if they would go into the Elizabeth River for us the next year when a bloom was initiating. And sure enough, we could see now we've added the Elizabeth, and here's the Lafayette River here. And you can see that the Lafayette and Elizabeth River light up first, and then they come out the James River mouth and across the James River, and then they get smeared up and down the James River. And we actually did a model at that time. We were doing this with a particle tracking model, and sure enough, that's exactly what happened if we seeded algal cells in or particles in the water in the Lafayette River here. They went out the Elizabeth River, then there's a stationary eddy that sits there, and then the tidal circulation just sort of smears it up and down the estuary. And I'll show you some model results later because we've extended that model to the coastal ocean. But again, this is just another year showing the data in a different way. We didn't interpolate this time. And again, you can see in 2009, same thing happened, is the Lafayette River lit up first, and then it went out into the the polyhaline part of the James River, and then out into the meso and oligohaline area. So we were convinced, and so we found that transport mechanism, and we found an initiation site. We did simulations using the VIMS HEM3D particle tracking model, and we did these dye simulations, and they simulated exactly what our populations were doing. And so it suggested that Margalefidinium was being transported to the James River, and the source or the initiation site was in the Elizabeth River. So, so we confirmed that. And so now let's look at our sampling program. Now we've added a time series site in the Lafayette River. And so now this is a site that we could study year round and learn about bloom initiation and what caused it. And that was the subject of our 2018 to 2022 EcoHAB grant that preceded our MareHAB grant. And so we did near daily sampling between 2012 and 2025. We actually sample year round now. We were only sampling during the summer months here. And now we've also added a station at the Elizabeth River, in the Elizabeth River at the Learning Barge, where there's also a data sonde located that's moored in the water. Okay, so what does this mean at our time series sites? Well, when we sample continuously, this figure is just showing a series of dinoflagellate populations that bloom. So in green, there's Akashiwo. In blue here, this was followed by a Scripsiella bloom. And then here's our Margalefidinium blooms. Oops, sorry, I didn't change the name. It used to be Cochladinium. And then the black is just the other dinoflagellates. But essentially what this figure shows is that we have this sequence of blooms that we showed with our data previously when we compiled all our temperature data. But so now when we see Scripsiella come, we start to look for Marge because we know it'll get intermixed at some point. This lower slide is actually just, that looks just all red. That looks pretty boring, right? That's just to show that the system is dominated by dinoflagellates. The red is dinoflagellates, and it's pretty much all summer long just dinoflagellates in the water. And FYI, dinoflagellates are sort of the Twinkies of the food web. If you want to think of diatoms as being the caviar, dinoflagellates don't make great food. But usually we'll see Margalefidinium start to emerge once the temperatures reach about 25 degrees C. So now we have this time series site monitoring, and we're capturing bloom initiation much better. And this is a figure that's in a submitted manuscript, and this shows our station at the Yacht Club is right adjacent to a Bay Program monitoring station. And so from that station, we look at the average chlorophyll plus or minus. This is the red dotted line is the 95% confident interval or confidence limit. And these dots are the different years, what we measured at our time series site with our high frequency monitoring. And we just averaged over the particular months at these particular places, just to show you the standard deviations. But now you can see that you don't see the blooms much in the monitoring data, the monthly monitoring, but now that we're sampling much more frequently, we're catching the blooms. So the addition of the time series has been great. So once we thought about that, we started to think about a conceptual model about what controls bloom initiation. And I've put in red under all my different arrows, different names, and these are people who have worked on these particular avenues over the years. And so we were examining meteorological forcing because that's important for nutrient inputs to a place like the Lafayette River. Temperature, we know that changes seasonally. We looked at other physical forcing from flooding and runoff. And then biological factors, we started to think about what limits phytoplankton growth, the nutrient supply. Dinoflagellates are also weird in that most of them appear to be mixotrophs. That means they can be both autotrophic and heterotrophic. Okay, so they can eat their competitors in addition to just outcompeting them. So that's a real advantage. Dinoflagellates also have complex life cycles, and so they make resting cysts that overwinter in the system. And so we think that's also very important. And so we started thinking about different sources of nutrients. There's runoff from flooding, there's atmospheric deposition, groundwater inputs we've now found are really significant. And then there are these biological forcing, and then the behaviors, the vertical migration. And so we know we started to draw this picture so that we could start to pick apart different aspects that we could work on and get a better handle on. And in 2021, or in the years before that, Eileen built a biological model of the blooms. And these were really important to parameterize a lot of these rates so that we could parameterize the model. And in particular, we examined with the model the role of mixotrophy, cyst dynamics, and that thermal window that we thought was so important. Okay, so here's the biological model that we built of the bloom cycle, and Eduardo drew this pretty picture with the Pac-Man here. But essentially, it shows bloom initiation here, and we need to somehow get these cells to exist. And we think water temperature might be the trigger for that, and that there are available nutrients potentially at that point. During the bloom proliferation phase, rather, we think the growth could be primarily autotrophic. We speculated, we used the models we tried to parameterize the model to simulate what we saw in nature, but we also tried to do experiments with the model so we could see what it took for the cells to reach the observed densities in different places. But during the proliferation phase, these cells were primarily autotrophic, but then we added about 30% heterotrophy to that. And then there was more dependence on heterotrophy later during the maintenance phase, and there was more nutrient stress because these things bloom when nutrients are depleted already in the water column. So we know that they're getting their nutrition elsewhere. And then there's the decline. We think that's linked with seasonal cooling, and then there's some trigger for encystment or going back to those sediments. In reality, it's really hard to make measurements of a lot of this stuff, okay? For initiation, we need to be sampling every day or two. And most monitoring programs don't do this. And this is hard on humans. So we, in the summer, have an army of people and try to set up a rotation so we can get samples nearly every day. We can measure chlorophyll on our sensors, but that doesn't tell us what the chlorophyll is comprised of, what actual cells are there. Most cysts, we have trouble detecting them. They just look like little round marbles, so they're not very distinctive. The nutrient concentrations are highly dynamic and vary over, hourly timescales, so really fast. And then, like I said, these cells are really complicated, and they're both heterotrophic and autotrophic. And there's actually no way to measure that in the field. We can measure autotrophy pretty well, but it's hard to measure the heterotrophy by a dinoflagellate eating another phytoplankton. So it's really tricky. So maintenance, a lot of times blooms persist and we don't always know why. They get transported places and they just seem to be, like I said, scrappy and be able to survive in a lot of places. So many studies do a single study in a single year and write the paper. And that's what I've learned I should have done, because from sampling in multiple years, there's no smoking gun to what's causing a bloom that we can put our finger on. We have several things that we think need to be aligned, but we have blooms in a lot of years, and there's no one thing that we can count as the definitive trigger. Encystment is very hard to measure. One of my students, Eduardo, he built a cyst trap to try to measure that. But we think cooling temperatures, which we can measure, we think that is pretty important. But again, that's correlative. It's not necessarily causative. We know the bottom is important, and this is just showing some of the data from our daily sampling at our time series site. The top panel is temperature, and the warm colors are warm temperatures, and the bottom panel is chlorophyll. And again, warm colors, high chlorophyll. During this particular year, I like to show as an example because we had a record storm with 7 inches of rainfall and high winds. And if we look at our sensor data and these from our vertical profiles that we do every day when we go out there, you can see that the chlorophyll, the upper panel is chlorophyll, it just got washed out. Okay, this was the wind. And then nutrients in the surface, they increased some in the surface water, suggesting runoff could have been important. But the nutrients in the bottom water, this is a different scale. It's increased by a factor of 3 over this one. But there's a huge pulse of bottom nutrients. And so we think the sediments are a huge source of nutrients to these systems. But sediments can also, they introduce nutrients, but they could also be important for introducing the cysts to the water. So in this particular study, we had an REU look at the nutrient concentrations in the water after we actually took cores and put overlying water there, and then we stirred them for a while. And sure enough, you could see that the ammonium concentrations went way up. The chlorophyll concentrations increased by a factor of 3. And then we even got nitrification by the end of it, once the chlorophyll died down and they got probably warm and a little bit low oxygen. And sure enough, if you take vertical profiles before and after wind events, you can see this huge bolus of nutrient being released from the sediment after a wind event. And the cysts, the wind can also resuspend cysts. And these were some studies we did again with the cores in the laboratory. And the red, the pink and red, these are just 2 different temperatures, so don't worry about that too much. But the red is mixed cores, the blue is unmixed cores. And in the mixed cores, you can see the cyst abundance in the overlying waters gets a lot higher. And that's important. So we resuspended cysts. And then the same thing is true with dinoflagellate abundance. This student didn't identify, he was unable to distinguish between different species of dinoflagellates, so he just counted total dinoflagellates. And so you can see that after these sediments were mixed, we had dinoflagellates increasing in the overlying waters as well. So this seemed to be causative. And then we went out and did a fancier study with fancier instruments that we put on the water. And essentially we found that in the Lafayette River, prolonged southwesterly winds cause resuspension events. They cause wave orbitals to increase in size, and they can cause sediment resuspension. And here we deployed Rip Hale's Aquadop, and you can see this was a resuspension event here. And then shortly after that, we had increases in nutrient concentrations in near bottom waters. And then we had an increase in chlorophyll concentrations. So again, this is correlative, not necessarily causative, but it seems to show that there's a resuspension event, elevated nutrient concentrations, and then your chlorophyll A goes up. This was not during a time period that Margolepidinium was happening, unfortunately. So this was, I think Akashiwo was the dominant dinoflagellate in that particular one. But so this brings me back to the biological model is very important because we know this is so important. We know heterotrophy is necessary to achieve the observed bloom magnitude, but we can't measure it. And we know the thermal window determines the bloom timing. And we were able to do this by incorporating many of the things we are able to observe into the biological model that Eileen created. And so we made some progress. And now when we combine surveillance techniques, this is just our Margolepidinium abundance. The orange is when there's greater than 1,000 cells per mil, and that's our bloom threshold. That's based on how much chlorophyll is expected to be there then, and that's based on the impairment level for chlorophyll that's set for the James River. And the red is when Margolepidinium is greater than 10,000 cells per milliliter. So when we combine our HRSD data, we had HRSD start to grab samples when they were out and when they observed chlorophyll concentrations above 25, and they would bring us a sample. And so we can combine our James River cruise data and our Lafayette River time series data, and we get a better picture of these blooms in different years. I'm only showing up to 2020 here because I just didn't have time to update it for this. We also can see blooms with satellite surveillance. Again, this can't tell us what species it is, just like the data flow and the chlorophyll measurements can't tell us what species it is. But if we can learn more about when things occur, we can guess. Or when we get these images, we try to get out and get a sample. But this is during a bloom in 2020, and you can see this is a Margolepidinium bloom, and you can see it's very apparent. You can see from these satellite images, we don't always get a satellite image every day, but you can see the bloom here, and it's even coming out the bay mouth, and that's part of the estuarine circulation. But again, we have to go out and get samples to be able to tell what this is. And the data flow, again, we combine these with cell counts. And again, we can see this is the 2020 bloom again. And we can see this bloom. And this bloom in 2020 was a very protracted bloom, and it went all the way out to the ocean front. They closed beaches. And our particle tracking model is now improved. And where is it not going to let me play it? Oh, I love this animation. I just checked it too right before we started, and I reloaded it. Okay, I guess it's not going to see it, but essentially this shows we, during this particular year, this bloom went to the ocean front within a week. And sure enough, based on the hydrodynamics in the particle tracking model, this bloom wound up just sort of swelling, boinging around back and forth, and made it all the way out and all the way up into this part of the bay as well. So, yeah, so sorry I can't show you that animation. Boy, and I'm so mad because wait, there it goes. And you can just see it really, the time period was right. There's the days here, and this bloom just swelled and transported down the coast. There's the big part of it coming down the Atlantic coast. And people did not like to see that at the beaches in July and August. Okay, so what we've learned, it's complicated. We have identified hot spots and hot times for blooms initiation. Estuarine transport determines where the bloom goes, and the bottom is important for the introduction of cysts and nutrients. And the biological model also needed the sediments and the cysts to be introduced and needed mixotrophy and a high degree of heterotrophy actually by the cells. During maintenance phase, it was up to 70% heterotrophic to get the bloom's size we observed. So, but what about some of those interannual differences? And reaching some temperature or salinity threshold is not sufficient still for predicting a bloom. And the wind and meteorological forcing can sometimes help. And mixotrophy, like I said, is needed to explain the bloom magnitude, but there are other things that happen. And this is the subject of a whole nother talk, but I'm just going to show you a couple of slides here about the heat waves, because this is was inspired by my COVID garden when everything died. And in 2019, well, we had a heat wave. This is chlorophyll concentration. These boxes on here are when we had bloom concentrations of Margolophodinium, okay? And this is temperature over here. And again, warm colors, either high numbers for chlorophyll or high temperatures. And this is 2019, 2020, and 2021. And it seems like the timing of when it heats up and the rate at which it heats up is really important. So in 2020, when we had that massive protracted bloom, we had high chlorophyll here. We had a heat wave, but not right when the bloom was initiating. Okay, so the water was hot, but then it cooled off. There was something happened there. So, and then we got the massive bloom. In 2019, when we did not get a bloom, the bloom was trying to initiate right at the outset of a 10-day long heat wave. It was really hot. It was over 32 in the water temperatures. And in culture studies, we've shown that Margolophodinium doesn't like to grow much over 32 degrees. And so we think initiation got nipped in the bud there. In this particular, in 2021, again, we had a heat wave that occurred later, but we had, again, another massive and protracted bloom. Not as much at the time series site, but the transport, it had transported away from the time series site, and so persisted in other parts of the James River and the lower Chesapeake Bay during 2021. But so now we think that the timing is everything. And again, this is, if you had to squint and you didn't think the colors looked just right on the other, this is showing the average, again, from the Chesapeake Bay program climatology, and 1 and 2 standard deviations from that. And then this line here, the different colored line, is the temperature record from our data sonde in 2019 up here, 2020 here, and 2021 here. And here you can see it better. In 2019, the bloom initiation was right at the outset of this massive heat wave that lasted about 10 days and above 2 standard deviations above the long-term average. And then it tried to initiate again in September, and it got cool right after that. And so it was probably below what it wanted to be. But in 2020, when we had the big bloom, we had a little bit of a heat wave here, but it was at the decline of it when the bloom initiated, and then it kept going. And in 2021, the heat wave wasn't until the bloom was well established, and we were okay. Interestingly, in 2024 and so far in 2025, we haven't had blooms. And there's been massive heat waves early in June and July in those years as well. So it's sort of consistent with this view. So I need to get this done. So now we've refined our conceptual model, but there's still a lot to learn. We are trying to do some experiments again with the model, looking at climate variability and change and looking at the heat waves. We've learned more about mixotrophy could be a key biological factor. We've learned lots of different things, and that cis dynamics are important as well. So we have started doing this, but we're still working from this and trying to do more. So now what? This was the point of the talk. We're trying now to we're at a stage where we can start to put all these pieces together to try to build a forecast for a HAB. So we can warn people when HAB is likely to happen. The fishing stinks when there's a HAB. So people that are doing fisheries do not want to go into bloomy waters. If you're doing aquaculture, you might want to close down your intakes because you don't want to choke your larval organisms, because Marge will kill them, as will Alex. And so now we're in the second year of our MARHAB grant, where we're trying to get a predictive understanding of these blooms so that we can forecast them. And so we have many different observing platforms, but many can't tell us what the species is that's blooming so that we can determine if it's harmful. We have moorings, we have satellites here that are indicative, we have data flow over here, but what we need is more observations. And so for that, we need to engage our stakeholders, and we're trying to build a network of imaging systems across the region so that we can start to collect samples when we see something, maybe from a satellite or a data flow, to where we can get a rapid turnaround to identify this species and to get a warning out to systems. And so we need more observations that are species specific. So here was our dilemma, okay? There are trade-offs between high frequency data collection. So microscopy is very laborious. It's so much work. And there are particle imaging systems. This is an imaging flow cytobot, and this is a flow cam. We have one of each in our lab, but these cost $185K. These cost about $150K. California has a network of IFCBs monitoring its coastal waters, but the state of Virginia, I'm guessing that they're not going to buy 7 or 8 of these instruments. And besides, where would you put them? It's so heterogeneous in the lower bay. But essentially, there are networks of these out there to try to detect HABs. There's another network in Texas, and they have these IFCB networks, but they're so expensive. And so we started looking at during COVID, we built our first planktoscopes, and we built them according to directions, using parts we could buy on Amazon. Now they sell kits in France for them. So when we bought off Amazon, the first time it cost about $2,000. And then the second one, because we already had the tools, only cost about $1,000 to build. They're selling the kits now for about $6,000. And we've proposed to put 30 of these planktoscopes around the bay in the hands of users who will run samples for us. And then, and I'll tell you how that works in just a minute. But essentially, these are low-cost imaging systems that you build yourselves, okay? And so what they look like is this. This is the schematic for it. It's all run off a Raspberry Pi. And then there's a little camera. There's a little pump up here. Well, they have it here with the that pumps the water through a flow cell past the microscope and the camera. So there's light, and there's a camera lens, and then there's a microscope objective here. And so when a pulse of water comes by, it stops the flow. You program the computer to tell you when you want to stop the flow, and it takes a picture. Okay, it takes a picture of everything like you would look at under a microscope. So it's going to have lots of cells in it. But essentially, you pump in the water. This is what it looks like in reality now, the newer ones. And essentially, you collect the water sample. You put that water sample into this syringe here. That then you use this pump to pump through your flow cell, which is down through there. And so you connect this planktoscope to a computer, or you can even use your phone. And then you have to enter the metadata for like what your location is, your sample, what your sample is, and then you essentially run your sample, acquire the data. And like I said, you're going to get a picture of everything under the microscope. I'm holding my hands in a little circle. So everything in that field of vision. So what you have to do is those more expensive imaging systems, they take a picture of every particle in the water as it goes by. It triggers a laser. And so for this, you're taking a picture as if you were looking under a microscope. So you have to cut out each cell in that field of vision. Okay, so that's called segmenting. And there's a computer program that will segment each image. And then you have to download those images and metadata. And then you have to find a platform in which to annotate all those images. The IFCB has its own platform. They are IFCB dashboards. And then the FlowCam has its own proprietary software for doing these annotations in. We've elected to go into Ecotaxa, which was developed by a French group for Terra Oceans. And it's an open source platform that can store the images, and you can annotate right in there. And the nice thing about it is you can annotate, say, about 50 of your images and put them where they belong according to the species. And then you can hit a refresh button, and it will annotate your entire database. And it will get a lot wrong. Okay, it'll get a lot wrong. So then you have to go in and move the things to their correct bins. And then you might do that for another 50 or 100 images, and then you hit the refresh again, and it just keeps learning. So we're trying to train Ecotaxa initially to recognize any of our HAB species that we see in the Chesapeake Bay. And so then we annotate and classify the images, and we use trained taxonomists to do that. And like I said, we use machine learning to continuously update those classifiers. And as we add more species and get new images, because the other thing with imaging systems, it's not like a microscope where if something is sort of two-dimensional, it's gonna sort of lie flat, and you'll see cells mainly in one particular orientation. When you're running things through these imaging systems, you might see them in all different orientations. So you have to get better at recognizing what it looks like if you're looking at it top down or from the side, or any particular orientation it's in. But it's a really cool process. And we've had 2 workshops now, and we're planning our 3rd, where we have invited the users here to ODU. We use the teaching lab, and we have them finish the build of their kit. We realized it wasn't good use of their time to build the entire thing. That would take too long. And so they just finish the build. It comes in a nice little Pelican case that you can keep it stored in, or if you want to bring it to the field, this is the operational planktoscope that you have. And then you run the samples and collect those images. And remember, I told you this is what an unsegmented image looks like. So when you take that picture under the microscope, and then these are the segmented images. And this is Margolefidinium. And we've put them in a particular classification within Ecotaxa. So we have to annotate and train Ecotaxa to do the job. Okay, we also, as part of this project, not everyone wants to deal with a planktoscope. And that whole workflow, we found that it's a bottleneck. People, sometimes the segmentation process, because that can take a while, or uploading to Ecotaxa as there are software updates, there are those issues. So we're also deploying sampling kits for users to collect samples and bring them back to ODU or VIMS. And these look something like that. And we have them deployed with quite a few recreational anglers, as well as some commercial fishermen as well. So that helps us enhance sample collection. A new development that's just happened last week, as a matter of fact, is the NOAA personnel. This is Chris Holland and Shelley Tomlinson, does our satellite support. Chris works in the Beaufort lab at NOAA, and they brought up a HAB scope, which is even cheaper than a planktoscope. It's only about $250, and it's an actual microscope. And they get 3D printed phone cases, and you put your phone on that case. You have to tell them which model phone you have. So we have a couple of them here now. And you put your slide under the microscope. And this is actually more satisfying for the users because the planktoscope, you can't really see the cells very good. The user can't see them. And so, especially when you have citizen scientists that are doing things, they want to see something. And so here, these are two chains of Alexandrium that we can see right there on your phone. And essentially, they're now training AI on the Chesapeake Bay systems. They have, these are already, they called them something else, and now I'm spacing on the name of it, but they have them functional, an operational system in Florida for Karenia, because Karenia has a really weird swimming behavior. So they actually built the AI around its swimming behavior. Some of our dinoflagellates are a little different and would be hard to distinguish because we have lots of different dinoflagellates. They're only worried about Karenia down there. And so we have to train that AI by getting lots of videos. And so we have this, and we're trying to get them to a couple other people so we can start training the AI so that this could potentially be operational shortly. And I think this may be the real future of it, because they have all these alert systems in place already for Florida when there's a HAB. So, we could take advantage of what they already have in place with the HAB scopes. So hopefully we can get some more of these, get that AI trained, and maybe then this will become part of the lexicon. So where we're at with the project now, and I'm winding down here. So we have 16 planktoscopes deployed with users in the field, and there are a variety of people. We have them at schools, we have them at agencies, we have one at the aquarium, we have a couple retirees that are really active users. And so we have 16 deployed. We want to have 30 deployed by the end of the project. So by 2028. We're annotating images in Ecotaxa. What we have them do is we have them upload to a Google folder if they don't feel comfortable, if the users don't feel comfortable in uploading to Ecotaxa, and then we take them from the Google folder, segment them, and put them in Ecotaxa. So we have a variety of things depending on the comfort level of our users in the field. We have the biological models. The modeling is a key component of this, and we've coupled that with the hydrodynamic model. And by we, I mean Eileen and Pierre and Margie at VIMS. And so they've coupled that with that. And then we have the working particle tracking model that they're now at NOAA, they're seeding with some of the satellite information of where it's at. And then they're using the satellites again to correct their tracking model. So a lot of those things are in place. We're really getting there. The forecasts are about there for these use a statistical model, and that uses environmental data. But again, to make sure all of these are correct and it's the correct species, we need those data on phytoplankton abundance. Environmental data are easier to get because we can do those with sensors and sondes, but up to today, we've had to do microscopy for the phytoplankton. And we're trying to continue to expand our network of samplers. And like I said, and still looking at different alternatives to a planktoscope as well. Some of the challenges, well, just enhancing the data collection, getting the users to use their planktoscopes and feel comfortable with them. A lot of times during the summer when we have the blooms, it's also people's field season. And so it's busy. And so, yeah, but we need to continue to enhance our data collection. The annotation platforms, this has been a real complicated thing. I'm actually going to an ICES IOS workshop next week where we're going to talk about this. Right now, Ecotaxa is nice because it's a really Catholic platform and will take data from flow cams, from IFCBs, from planktoscopes, from microscopes, from everything. And that's why we like to use it. But it was developed by the French group, like I said, and it's been handed off to IFREMER, which is the French equivalent of NOAA. And so they will not want to continue to support every onesie and twosie user around the world forever, indefinitely. They haven't cut anyone off yet, but we're hoping we have a proposal going in to try to develop a US instance of Ecotaxa that would span the whole East Coast. And we're working with people at NOAA, Naracuse, and Syracuse, I think. So we're working on that. The other thing, the time it takes to train users can be daunting because they need to use it in the field. And so we've been having office hours for our planktoscope users so they can ask questions. Transmitting information in near real time. We're still not there for planktoscopes. And because our training, our taxonomic training still has work to do, we need to get lots of images in there to get our AI working really well. And so the HAB scope may have a response to this one. And like I said, there's an international effort to build common platforms for annotating and storing systems. It's actually someone from Germany wants access to all of our Ecotaxa files because, again, they're trying to standardize the taxonomy and re-examine the distribution of the information and distribution of HABs. It's really cool because if we could standardize taxonomy, that would be great because even right now, sometimes people from the Maryland part of the Bay and Virginia Bay identify things differently. So, that could, so having these common platforms will really help us expand our knowledge and understanding of HABs. And so these are a lot of the people we've engaged to be planktoscope users and be part of our team and that have been helping us. But like I said, it's still an ongoing project. And then I also want to thank all the sampling team over the years because it's been armies of people, and I don't have pictures of all the interns as well that I need to add that we've had interns from Norfolk State the last few years that have been really helpful, and undergraduate interns, and as part of the class I teach as well, they've been going out and sampling at the time series site. So anyway, thanks, and it's a work in progress, so I look forward to updating you.